So we know the typical cell has a lot of these different organelles. However, all of these organelles that I drew with the little red star, all these organelles are collectively referred to as the endomembrane system. So what is this endomembrane system? Well, first of all, we know we have the plasma membrane. And we know this plasma membrane is made out of these phospholipids. For example, we drew a phospholipid here. So for example, we have this bilayer membrane made out of these phospholipids. So this plasma membrane is made out of a bilayer of these phospholipids, these two layers of these phospholipids. But that's not just true for the plasma membrane. Also, the lysosomal membrane and the Golgi membrane and the endoplasmic reticulum membrane are each made out of these bilayer membranes. So every single one of these yellow lines I drew, for example, this yellow line for the Golgi and this yellow line for the endoplasmic reticulum, each of these yellow lines is made out of these bilayers, made out of these phospholipids. So the entire endomembrane system is made out of these phospholipid bilayer membranes. Each of these membranes are made out of these phospholipid bilayer membranes. So something that's a little non-intuitive is that the cell is kind of made out of two different compartments. First of all, we have the cytoplasm. So this black space is referred to as the cytoplasm. But we also have this endomembrane system that I drew in purple. And a way you can think about it is this entire endomembrane system, all these places that I drew with purple are all interconnected. So this endoplasmic reticulum lumen is interconnected with this Golgi lumen. How so? Well, you can imagine in this endoplasmic reticulum a vesicle pinching off. So we can imagine a vesicle pinching off. So if a vesicle is pinching off, we can see that the inside of this vesicle is continuous with the inside of the lumen of this endoplasmic reticulum. So then we create a vesicle, then that vesicle fuses with this Golgi, and when this vesicle fuses with the Golgi, we can see that this inside of the vesicle is now continuous with the lumen of the Golgi. So that way we can see that all this purple space is somewhat continuous. And then maybe another vesicle is released, but again, in the same fashion, we know the inside of this vesicle was somewhat continuous with the inside of this Golgi. And then when the vesicle fuses with the plasma membrane, we can see when this vesicle fuses with the plasma membrane, this inside, this purple, this endomembrane space is now continuous with the extracellular space. So also we can kind of think of the extracellular space as continuous with this purple, this endomembrane space. But the point is, you can think of it, one way you can think of it is the cell is kind of made out of two separate compartments, the cytoplasmic space and then the endomembrane space. And these two different compartments are separated by these phospholipid bilayer membranes. And something interesting to realize is there are different environments. For example, in the cytoplasm, we have a reducing environment. So in all this black space, the cytoplasm, we have a reducing environment. However, in this endomembrane space, generally speaking, we have an oxidizing environment. So we have these two separate environments that are again separated by this phospholipid bilayer membrane. And we also have different pHs. For example, the pH of the cytoplasm is generally speaking around 7.2. However, the pH of this endomembrane space varies. For example, in the lysosome, the endomembrane space has a pH of 5.5. However, in the lumen of the endoplasmic reticulum, the pH is around 7.2. So we kind of have these two separate compartments, these two separate components of the cell. So something else important to realize is we know sometimes we have a gene. And we know we can take that gene, we can transcribe it into mRNA, the mRNA leaves the cell, then that mRNA gets translated into a polypeptide. So we start translating this mRNA into some kind of nascent polypeptide. So we start creating this polypeptide, we start translating this polypeptide, and eventually we fully translated that polypeptide, and eventually we create some kind of polypeptide or protein. So this is how we create cytoplasmic proteins. If we create a protein or enzyme that needs to be in the cytoplasm, this is the way we do it. For example, maybe this was an enzyme for glycolysis. We know glycolysis occurs in the cytoplasm, so this is how we would create an enzyme that needs to be in the cytoplasm. However, what if we need to create an enzyme that needs to be in this endomembrane space? We know these enzymes and proteins can be very large. So if we create these large proteins, they can't just simply cross through this phospholipid bilayer membrane. So therefore, if we need to create a protein in this endomembrane space, how do we have a protein enter inside this endomembrane space? Well, the way we do this is, let's say we have another gene. Let's say this gene creates a protein that's supposed to be in the endomembrane space. Essentially what we do is we transcribe this gene to create mRNA. The mRNA leaves the cell, and then that mRNA begins to get translated, and as it begins to get translated, we create this nascent polypeptide with these amino acids. However, sometimes we get a very specific sequence of amino acids. For example, let's say we have the red, white, and blue amino acids. So let's say each of these different colors represents a different amino acid. So let's say we get this very specific sequence. If the cell sees that we have this very specific sequence, that tells the cell that this protein is supposed to be in the endomembrane space. So this very particular sequence of amino acids we refer to as a signal sequence. So this gene starts to get translated, it creates a few of these amino acids, which we refer to as a signal sequence, and this tells the cell that this protein is supposed to be inside the endomembrane space. So now the cell sees this signal sequence, and then the cell essentially takes this ribosome and these amino acids and kind of force them within this endoplasmic reticulum, this rough endoplasmic reticulum. So now you can see now that ribosome is now part of this endoplasmic reticulum, where now we see these amino acids that are being translated. So let's draw this a little larger. So again, this is our ribosome. This is the lumen of the endoplasmic reticulum. And again, this is our nascent polypeptide chain. And again, we know the rough endoplasmic reticulum is characterized by these ribosomes. These ribosomes, and this is one of those ribosomes that characterize this as the rough endoplasmic reticulum. But again, so we start translating this protein. And again, because of the signal sequence, it forced this ribosome into this region. So now we just continue translating this mRNA. So we continue translating, adding more amino acids, and then eventually we've completed translation. So now we've created this protein or this enzyme which is inside the endoplasmic reticulum. So now let's go back to our original picture. So now we've done it. Now we've created a protein that's inside this endoplasmic reticulum. However, maybe let's say this protein is actually, so we can create certain proteins that are supposed to be in the endoplasmic reticulum, so they'll just hang out in the endoplasmic reticulum, but sometimes we create other proteins that maybe need to be excreted outside of the cell. So how do we take this protein and secrete it outside of the cell? For example, maybe this is a beta cell, and this beta cell is producing insulin that it needs to secrete outside of the cell. So how's the cell going to do it? Well, again, the first step is essentially it's going to start developing a vesicle. So again, we can see it's going to develop this vesicle, which with this protein, and let's say this protein is insulin, that has this insulin inside the vesicle. Now the vesicle buds off, and now we can see the vesicle fuses with this Golgi apparatus. And when it fuses with the Golgi apparatus, now this insulin is now inside the Golgi apparatus. So again, like I said earlier, this all this purple region, this endomembrane space, is all continuous. You can think of it all being continuous. So now we have this insulin inside this endomembrane space. And then the Golgi matures. And then eventually what would happen is we'll take this insulin, it'll bind, forming a vesicle. And then remember, the inside this endomembrane space, once it fuses, is now continuous with the extracellular space. So now we can secrete this insulin inside the extracellular space. So this is referred to as the secretory pathway. And something else important to realize is within this Golgi endomembrane space is where a lot of post-translational modifications occur. Because again, we kind of have these closed compartments. So inside these closed compartments, we can go through post-translational modifications. Because again, we know that this polypeptide has already been translated. So this is a post-translational modification. And so within this Golgi, we can go through post-translational modifications where maybe we can take this protein and we can add carbohydrates. We can glycosylate it. Or maybe we can acetylate it, or we can phosphorylate it, or lipidate it, etc. So within this Golgi, the lumen is where we do a lot of our post-translational modifications. But now here's something that's really interesting. So let's say we created two separate proteins, and let's say they had the correct signal sequence to allow those proteins to be translated inside this endomembrane space. And then we know they go through this process. So now let's say we have two separate proteins within this Golgi. But let's say this particular protein is supposed to be secreted outside of the cell, while this particular protein is supposed to essentially pinch off to create a vesicle that eventually turns into a lysosome. So how does the Golgi apparatus know to take this protein and to secrete it outside of the cell? And how does it know to take this protein to essentially add it to a vesicle to turn into a lysosome? Well, essentially what happens is while these proteins go through this process, they'll get tagged. So maybe this particular protein happened to have an amino acid sequence that made it tagged with a certain component. So maybe this polypeptide got tagged with a certain component. However, maybe this polypeptide had a different amino acid sequence that forced it to be tagged with another component. Maybe this polypeptide was tagged with the monos 6-phosphate. But the point is, both of these different polypeptides have been tagged. And now, once they're tagged, essentially what they can do is they can bind to these membrane receptors. So again, this tag helps this protein bind to this membrane receptor. And then once it binds to the membrane receptor, then it pinches off to form a vesicle, which can now be secreted, and now the protein is secreted outside of the cell. However, this particular protein got tagged, which binds to this membrane receptor, which then pinched off to create this vesicle, which turned into the lysosome. And again, generally speaking, the way this works is once this protein binds to this receptor, then we essentially form a vesicle, and then through this kinesin-dynein mechanism that walks through this microtubule, essentially takes this vesicle and brings it to this part of the cell where then now the vesicle can fuse and now the protein can be secreted. But that's how the cell knows to take this protein to secrete it and to take this protein and to essentially bring it inside a lysosome. So something else important to realize is, let's say we have this protein that's part of the plasma membrane. So let's say this protein happens to be highly glycosylated, where it's highly glycosylated. So how do we create this protein that's part of this plasma membrane? How does this protein get here? Well, again, it's a similar idea. So let's say we have this gene. Let's say we have this gene that creates a protein that's supposed to be in the plasma membrane. So we take this gene, we transcribe it into mRNA, then that mRNA leaves the cell, and then that mRNA starts to get translated and starts creating this nascent polypeptide. So let's say this nascent polypeptide is made out of these particular amino acids. The cell will see these particular amino acids, and these particular amino acids will act as a signal sequence, and it will tell the cell that this particular protein is again supposed to be inside the endomembrane space. So now the cell will see this signal sequence, and then it will take this ribosome, and then it will add it to part of this rough endoplasmic reticulum. So again, now we've added it. So again, similarly, now we've added it to the rough endoplasmic reticulum. But let's say these particular amino acids essentially tell the rough endoplasmic reticulum that this protein is supposed to be part of the membrane, the membrane. So essentially, as it continues to get translated, it kind of gets forced inside the plasma membrane. Remember, this particular sequence told the rough endoplasmic reticulum to essentially force this polypeptide inside to become part of the plasma, to be part of this membrane. So now that this mRNA continues to get translated, and now we've created this polypeptide that's part of this membrane. So again, now let's go back to our larger picture. So now we've done it. Now we've created this protein that's part of this membrane. And it's hard to see this protein, so let's draw it in red just so we can see it a little better. But we know, so sometimes the proteins are supposed to be here. Certain proteins are supposed to be part of the rough endoplasmic reticulum. But again, remember, this protein is actually supposed to be part of the plasma membrane. So how do we take this protein and deliver it to the plasma membrane? Well, we already explained how we do this. Essentially, what would happen is where this part of the protein was, this part of the membrane will pinch off to form a vesicle. And now the vesicle will fuse with the Golgi. And when it fuses with the Golgi, now the protein is part of the membrane in the Golgi. And then the Golgi will mature, and then this part will pinch off to form a vesicle. Then that vesicle will fuse with the plasma membrane, and once this vesicle with this lipid bilayer membrane fuses with the plasma membrane. Now this protein is part of the plasma membrane. So that's how we create these plasma membrane proteins and how we deliver proteins to the plasma membrane. But remember, this particular protein was highly glycosylated. So essentially what happens is when this protein was still part of this endoplasmic reticulum membrane, we add this kind of template with a lot of these carbohydrates. But as we continue with this process, we modify these carbohydrates where we get rid of some carbohydrates and we add some other carbohydrates. So this carbohydrate template gets matured. And eventually what would happen is we'll remove certain carbohydrates, we'll add other carbohydrates, and then now we have the exact right carbohydrates. Because remember, we do a lot of post-translational modifications in this Golgi. So we're going to remove carbohydrates and add carbohydrates, etc. But eventually we have the right carbohydrates. So now this protein that's part of this membrane will pinch off and now can add as part of the plasma membrane. But something really important to realize is remember that this side of the protein that was part of this purple endomembrane space, so this part of the protein, once it binds, is now part of the extracellular space. So something important to realize are these membranes are kind of have a polarity where they have a side that's facing the cytoplasmic face, and they have another side that's facing the endomembrane space. And something important to realize is this side that doesn't have the carbohydrates. So the side that's always facing the cytoplasmic face will always face the cytoplasmic face. For example, we see this part of the protein that's facing the cytoplasmic face. Once it gets to this region, this side of the protein is still facing the cytoplasm. However, this part of the protein that's facing the endomembrane space, once it fuses, is now facing the extracellular space. So I know it's a little confusing, but the point is, the leaflet, the phospholipid bilayer leaflet, or essentially the face that's facing the cytoplasmic face, will always face the cytoplasmic face. So we kind of have a polarity. So now let's talk about lysosomes. So again, these lysosomes are part of the endomembrane space. And these lysosomes have a lot of hydrolase proteins, a lot of hydrolase enzymes. For example, we have glycosylases and nucleases and peptidases and lipases, which are all hydrolases. And we know what these hydrolases do. For example, if we threw in a carbohydrate in this lysosome, we know this glycosylase will chop it up. This glycosylase enzyme will chop it up. If we throw in a protein in this lysosome, we know this peptidase will chop it up. If we throw in a lipid, we know the lipase will chop it up. And if we throw in a nucleotide or a polynucleotide, the nucleases will chop it up. That's what these hydrolases do. They chop up biomolecules. So this lysosome, if we throw in proteins or biomolecules here, this lysosome will chop it up. So we know these lysosomes have lots of different functions. For example, they can function in autophagy. And again, I talked about autophagy in a previous video. I have a link of that video below. But these lysosomes can also have other purposes. For example, let's say we have this extracellular component that we want to digest and chop up. How do we do that? Well, first this extracellular, let's say it's a protein, will first bind to the plasma membrane. So it'll bind to the plasma membrane. Specifically, it'll bind to these plasma membrane proteins. And we've explained how those plasma membrane proteins get there. So first it'll bind. And now what's really interesting is once it's bound, we'll recruit these proteins. And there's lots of different types of proteins like clathrins and calveolin. But the point is, we recruit these proteins that have a curvature. You can see that they kind of look like little C's. They have curvature. And essentially, once these proteins, we bring these proteins that have curvature, essentially they induce a curvature. These proteins have curvature, so when they bind, they induce a curvature, which allows this vesicle to be endocytosized. So now we endocytosize this vesicle, which has this protein that we want to chop up. So how do we chop up this protein? Well, again, essentially what we need to do is we need to deliver hydrolase enzymes inside this purple region so we can chop up this protein. And we explain how we do that. We explain the whole process, how we have some kind of protein. So let's say this particular protein, and we explain how these proteins get in this endomembrane space. So let's say this particular protein happens to be a lysosomal hydrolase. So we know that this lysosomal hydrolase goes through this whole process. Then at some point, this lysosomal hydrolase will get tagged with the mannose-6-phosphate. So once this hydrolase enzyme gets tagged with this mannose-6-phosphate, it will essentially bind to this protein, this membrane protein. So once it binds to this membrane protein, then it pinches off as a vesicle. So now we have this vesicle with this lysosomal hydrolase. And then eventually this vesicle can fuse with this vesicle, and when we do that, we essentially release this lysosomal hydrolase. And again, so now we have a lot of these lysosomal hydrolases. And this isn't just going to occur once. There's not just one vesicle binding. There's going to be lots of different vesicles binding with different lysosomal hydrolases. But now they bind, so now we have something called an early endosome. So this early endosome is that original vesicle that had this pink protein. And once it's fused with some of these vesicles that had those lysosomal hydrolases. So they fuse, forming this early endosome. However, in this early endosome, these lysosomal hydrolases are inactive. So they are not active. So in the early endosome, these lysosomal hydrolases are inactive. So how do we activate them? How do we activate them? Well, essentially what we need to do is we need to create another protein. And again, we explain how we create these proteins part of the membrane. So again, this protein, again, forms a vesicle, and then the vesicle fuses with the Golgi. And then this protein that's part of the membrane can pinch off, forming another one of these vesicles. And remember, these vesicles can have lots of different things in them. They can have this red membrane protein. They can also have these lysosomal hydrolases. There can be lots of things in these vesicles. But essentially, these vesicles will fuse. And when the vesicles fuse, the membrane of this vesicle becomes the membrane of this early endosome. And remember, in the membrane of this vesicle, we had these red proteins, these membrane proteins. So now this early endosome has these membrane proteins, which again we created in the rough endoplasmic reticulum. And these particular membrane proteins, let's say they happen to be hydrogen ion transporters. So essentially what they do is they pump hydrogen ions inside this vesicle. And again, they'll do that with the use of ATP. But again, we'll have these ATPases, which are essentially pumping in hydrogen ions inside this vesicle. So as we pump in these hydrogen ions inside this vesicle matures into a late endosome. So then we'll continue pumping these hydrogen ions, and as we pump these hydrogen ions, the pH of this late endosome will decrease and will create a more acidic environment. And eventually we'll pump in so many hydrogen ions that we drop the pH to a pH of around 5.5. So once we reach this low pH, this very acidic environment, then these lysosomal hydrolases activate. So these lysosomal hydrolases are activated by pH. Low pHs activate these lysosomal hydrolases. So now these hydrolases are activated, and now we've created a fully mature lysosome. So now that these hydrolases are activated, they can start digesting this protein or whatever it was. Because again, we know we have lots of different hydrolases. We have glycosylases and lipases. So whatever biomolecule this was, now we can start digesting it in our lysosome. So this lysosome, again, is part of this endomembrane system. So all these organelles are referred to as the endomembrane system.